in the Brett Kagan's like chief scientist for cortical labs.
Okay.
And they so I think we covered this paper, which is just Brett Kagan by himself, perspective paper, where the big image that's important is this I don't know if it's going to come out in the video, but this like distinguishing organoid culture from what he's going to call bioengineered. It's just not coming through on that.
Well, I remember that figure. I don't. Is it this paper or another paper? I think it's another.
No, it's a different paper.
Right.
I mean, I think we talked about this one, which was just talking about these kind of like I loved it because it was going over kind of like the organoid path versus something that could be seen as the 3D bioprinting path.
Right.
And sorry, that alarm. And this one is with so he had a paper at the beginning of the year. I mean, I think it was sitting in bioarXiv maybe before that, but he had a paper this year that was like, how to set up your synthetic intelligence lab. And it was like a tutorial paper, but like detailed, like 60 pages plus a supplementary section, with the biological equipment that you need, the reagents that you'd need, the kind of cell culture work, the kind of every aspect, again, in my opinion, as someone been searching around for this kind of information, it was a absolutely needed, it was filling a hole, right? Where like nobody really had a textbook on this, and cell culture work in general. It's also lacking a lot of that. I mean, in the sense that like it's taught via practicum,
right?
And but it means that setting up your own space is still left as this kind of like it's transmitted from one generation to the other via the oral tradition or something like that. So that was a nice paper. One of the things that was funny about that paper, or the oddity to me about it, was that it had the kind of people that you'd expect, like Brett Kagan, and a guy on this paper, I'm blanking on his name right now, but like Mohammed
Mostaja Raji or something like that.
Yeah. Who's a brain engineer, right?
Okay.
So he's a UCSC guy, not Kate's PI, but somebody she's worked with for sure. But then there's all these Rochester Institute of Technology people, right? So that was on that's on this original tutorial paper, and they're all on this one too.
Okay.
And it's interesting because it's a NeuroAI paper. I mean, it's definitely like trying to focus on that, and I mean, I can't say I'm very happy with the term NeuroAI.
Right. It's
Or, you know,
kind of a buzzword.
They continue. I mean, they're doing what I would consider NeuroAI, but nobody else does.
Right.
And so that just makes the term confusing. But it's interesting because it starts to get into kind of like some of the neuromorphic like. You know, because they want to talk about how to use biological neural networks, you know, in ML, they start to at least discuss, you know, the history of neuromorphic computing and things like that, which again is like obviously have a, well, no, not obviously, but at least in implementation have a digital form, right? Even if you're yeah, just that. And so again, interesting, like makes a little bit more sense that Guy Wang's group would be involved in that, because when I look him up, he's like a medical imaging researcher.
Yeah.
You know, he's like, I think he's somehow famous in the kind of CT world. Anyway, but I'm sure he has a lot of computational people. So somewhat relevant to Emily Sherman's Inspire program, as well as somewhat interesting for, excuse me, somewhat relevant to Josh Bongard's. So he also had another talk this week, the computational philosophy group, this et.al group.
Okay.
And you know, so I mean, this is just on the heels of excuse me, okay, this is on the heels of the Foresight meeting with Michael Levin and Josh Bongard.
Okay.
But this computational philosophy group one was very much centered around his polycomputational paper from I think 2023. So this is the
You're talking about Josh Bongard? Oh yeah, Josh Bongard. Okay.
Yeah. It's Josh Bongard. I mean, you know, it's like he was definitely going to talk about anthropods. But it's not really his thing. I mean, you know, he seems, and certainly if you go through his course, like he's the guy's much more, you know, simulations
right.
of yeah, you know, soft robotics kind of, and he can relate that to anthropods, but this polycomputation paper is very much in the, well, it's related to some of the papers you've presented on unconventional computing, you know, especially around kind of mechanical processes.
Okay, yeah.
I can't think of any actual titles right now, but you've definitely talked about some of these.
Yeah, I can't remember which ones they are, but I remember the polycomputation paper, and that was where they were like programming, almost like programmable matter, kind of in that direction.
Yeah, I mean, that's definitely one of the ways he was going, and you know, he had, I mean, so it's maybe if Jess joins later, he can say more, because Jess, when Josh had already started, Jess was like, "Oh, this sounds like he's going the Brainberg vehicle route." And then he totally put up pictures of Brainberg vehicles, but actually came to overlay metamaterials onto Brainberg vehicles, or it was that was kind of his contribution, and where the kind of gates that he was creating could be those mechanisms of those early vehicles, right? Anyway, it was an interesting discussion. I was kind of I think I said in the Slack, I was kind of on a deadline for somebody's letter, but what I was interested to see was what kind of analog computing examples he would bring up, and especially if any of those would be relevant to brain modeling.
Yeah.
I mean, almost of the sorts of brain modes, right? Like although they didn't, they're usually doing these kind of decompositions as opposed to like the kind of I really wanted him to try and connect some of his work to like Earl Miller's work, and the kind of people who present at the Wave Club. Do you know that seminar series?
No, I've never heard of it.
It's Wave Club. So you're in luck. Next week, the Wave Club from this week, because it's the first. So traditionally, I think it's like the first Wednesday of the month.
Okay.
Or something like that. What's the 14th? No, it's the first Tuesday. Must be the first Tuesday of the month.
Okay.
And it's Bard Ehrmantraut.
Oh, okay.
Right, is like, right, so you can already get a sense of like, what kind of stuff it's going to be, and it's mostly, it definitely has. I mean, I don't know if this is what it's set up for, but it's like it's mostly electrophys people who are looking at traveling waves.
Okay.
In brain activity. So it was like Earl Miller, although I mean Earl Miller's definitely spoken there. I feel like the last time Earl Miller spoke, though, it was who's that guy at University of Maryland, like Louis Pessoa? How do you pronounce his name?
Yeah, Luis Pessoa, I think.
Louis Pessoa, like the entangled mind, entangled brain, or something.
Yeah.
Right. So he hosted Earl Miller most recently, like that's why it was on my mind, or
Okay.
that, and specifically it was like Earl Miller was talking about, by having by having a complex spectrum of frequencies active, that you could be doing something that allowed you polycomputation.
Okay.
And and it was also how they're setting it up to expand. I mean, if the neuromorphic people, right, are making kind of like leaning on these brain metaphors, or inspiration,
Yeah.
right, like a lot of the analog computing seems to be kind of like doing the same thing but drawing from quantum computing, that that somehow this is a mechanism that will actually allow a lot more parallel computation, right? Which, parallel, poly, I mean, and but I never get enough detail, but they never go into the kind of detail that neuromorphic computing people will go into in terms of actually trying to make a simulation, or, God forbid, an actual practical implementation, right?
Yeah.
It's more these kind of these nods towards these ideas, as opposed to actually like putting together examples of them. And to some degree, the Bongard work was the same thing, but like it was like the magic was the metamaterials.
Yeah.
Where it's like and then metamaterials, and like that'll, like, okay, do we have any examples? Any of these, practical, even at a kind of organoid intelligence level, right? Like, I mean, reservoir computing is a thing,
Right?
Like, you can do it with non-living matter, of course. But anyway, along aside, I mean, but relevant to the Emily Sherman stuff, or like that DARPA program. The sad thing is that she's finishing up at like her time at DARPA, and you can't blame anyone for not wanting to be a part of the federal government right now.
Yeah.
So she was probably the best advocate for somebody to be doing organoid intelligence. I mean, she was the art fellow who, was going to be the closest to that. Anyway. But the Giwang, or this latest Brett Kagan paper tries to be tries to kind of gather a lot of the references relative to neuromorphic computing, and, the algorithms that have been used by biological intelligence, synthetic intelligence people, and so if we hadn't covered it, I just thought that would be a good one to mention as another,
Yeah, but why don't we go over that paper?
Sure. Yeah, and it's got some good figures like the other one too. It's still an archive paper, but they're obviously planning to to get this out with the same kind of development as these others that they've done, which have been great. I mean, that tutorial paper again is like, absolutely the best reference on the subject.
Yeah. This is the paper, A Computational Perspective on NeuroAI and Synthetic Biological Intelligence. And the NeuroAI term, of course, is kind of a buzzword. I mean, we're using that as a stand-in for anything that isn't like neural networks, I guess, or, something similar. This is on the arXiv from, I guess, this week. And the authors here, Brett Kagan, Ji Wang, Mohammed Mustajo, Raji, and a couple other people here. So then zoom in here. So,
thank you.
they're from Cortical Labs, RPI, Santa Cruz. That's where we're talking about. So
most of them,
Oh yeah. So NeuroAI is an emerging field at the intersection of neuroscience and artificial intelligence, where insights from brain function guide the design of intelligent systems. A central area within this field is synthetic biological intelligence, or SBI. So this is the organoid stuff, which combines the adaptive learning properties of biological neural networks with engineered hardware and software. SBI systems provide a platform for modeling neural computation, developing biohybrid architectures, and enabling new forms of embodied intelligence. And so this is a review where they organize the NeuroAI landscape into 3 interacting domains: hardware, software, and wetware. So this is hardware being silicon hardware, software being some programming language or some machine language, and then wetware being the like organoid work and potentially like brain circuits. We outline computational frameworks and highlight recent advances in organoid intelligence, neuromorphic computing, and neurosymbolic learning. These developments collectively point towards a new class of systems that compute through interactions between living neural tissue and digital algorithms. So this is, kind of a nice vision for things. So let's go through the paper. This is kind of selling NeuroAI as a paradigm shift and how this is just kind of the way that we need to go, combining the insights of the brain with artificial intelligence. What that means, of course, is up to the research group that's doing it. So, this is a multidisciplinary effort. It requires people to think, both in terms of biology and in computation. And there are all sorts of applications. They always have to highlight the medical applications, the therapies and other types of cognitive aids and things like that. And they kind of get into, the history of tissue culture, I guess, and then kind of walking through that. Let's see.
It's I mean, it starts with tissue culture, but it's like, it's specifically about neurons.
Yeah, they get into the Lancaster papers here, Madeline Lancaster.
Yeah, but like neurons, and then MEAs, and then kind of like how to build, how to start, do stem cell work, and then organoids.
Yeah. Okay. And then, of course, computational science, they go through kind of some of the history of that and kind of going from the early years of computation to reinforcement learning to deep learning and other types of neuroscientific theories like global workspace theory, integrated information theory, predictive coding, and active inference, and how those can be useful in this context. And then, of course, I talk about neuromorphic hardware driving this kind of spiking event-driven architecture of biological neurons. Okay, so here we examine the emerging field of SBI and NeuroAI with a focus on its computational foundations. We review key technological conceptual advances, algorithms, and architectures that connect biological and artificial forms of intelligence. By highlighting these integrative efforts, we provide a comprehensive framework for understanding the current state and future directions of this rapidly evolving field. So they get into biological neural networks, or BNNs, and this is the foundation of what they want to do. They talk about the complexity of single biological neurons. So ANNs are sometimes described as brain inspired, but they're extremely simple in terms of their function and operation. It's basically a unit that gives out, spits out weights. And that's not how actual neurons work. How actual neurons work, of course, is much more analog and has a lot of different parameters that you can tune. They get into dendritic trees and dendritic computing. So dendritic trees act not as single summing points, but as distributed filter banks, as shown in figure 1. Each branch approximates a leaky RC cable with distinct membrane resistance, axial resistance, and capacitance. Synaptic inputs undergo location and time-dependent attenuation, with distant signals arriving more weakly and slowly than proximal ones. So this is a tree here that they show, and they show kind of there's a circuit at each node where they have different electrical parameters. Membrane resistance, axial resistance, and capacitance. So you're modeling this physical system within each node, and then you're summing those together, but after they've been processed. So this is like a dendrite where you're getting sources from different places, and it's being filtered into the neuron. Okay, so this just shows the dendritic branches here. And it's just underscoring that it's not at all like an artificial neural network. It has the structure that is not really accounted for in our typical networks. Even in something like reinforcement learning, you don't really have this kind of structure. Although in biological reinforcement learning, we kind of assume the structure. Actually, biological reinforcement learning is a lot about dopamine signaling. So there's a whole different area there. But this is kind of like, there's, of course, the area of dendritic computing where people are doing a lot of, kind of pioneering work in that area. And they're kind of using the dendrite as the main unit of information processing. Okay, so then there's this key differences between BNNs and ANNs, biological and artificial neural networks. So their focus here is on the properties for each one. They focus on training data requirements. So in BNNs, we have evolutionary pre-training, innate threat detection, and feature extraction. So that's the way they kind of state that. So in other words, like you have things that are innate or encoded in, for different animals, like if you have a fight or flight response, that's evolutionary pre-training. Innate threat detection and feature extraction are things also that are innate that are kind of analogous to pre-training in an artificial neural network. And then, of course, you have large data sets in artificial neural networks. One thing they didn't point to here is that biological neural networks don't require a lot of quote-unquote training, where the training, rather, is not, you don't have to learn, you don't have to be exposed to the environment as much as possible to learn things. I mean, you get exposed in development to things. We've talked about that before, where development is about being exposed to a lot of things, but also like being focused on different things and playing with them and seeing how they behave. So it's a very different type of training than like what you get in artificial neural networks. So there's that difference too, but I'll put that aside for now. The second property is energy efficiency. So biological neural networks operate on a few watts of glucose, and artificial neural networks have high computational resource consumption. So this is like where we have our huge data centers that require a lot of energy versus like a human brain that requires basically food and water. And then, that's enough energy to produce, what the brain does. Then the third point is intra-network communication. So this is where we have the focus on synapses, complex biochemical interactions at synapses. You get all sorts of signaling at synapses, as opposed to artificial neural networks where we have simple mathematical operations such as addition and convolution. So, in the biological brain, you have these, biochemical signals, you have binding to receptors, things like that. In ANNs, and then you can maybe characterize those with mathematical operations. In artificial neural networks, they're basically, the operations are the entire sort of interaction between neurons. So it's all it's all kind of condensed into addition or convolution or multiplication or something like that. The learning mechanisms. In biological neural networks, we have self-organization, synaptic plasticity, and predictive coding. In artificial neural networks, we have gradient-based backpropagation. So that's one. This is kind of the example to use from artificial neural networks. The idea being is that artificial neural networks are optimized. They follow a gradient to learn things. In biological neural networks, basically learning is, I don't know if there's a gradient. People often refer to learning gradients, but they're kind of informal structures, and really the focus is on like synaptic plasticity, the predictive nature of the brain, and then self-organization of networks. So it's a very different sort of paradigm there. And so then the next one is systems architecture. So that's biological neurons with integrated storage and computation. And then in artificial neural networks, we have silicon-based transistors, von Neumann architectures, which separate memory and processing into two components. Whereas in biological networks, those things aren't necessarily separated, or if they are, they're in a network that interacts with each other. So it's a very different architecture. Emergent properties is the next property, and this is where we have, in biological neural networks, spontaneous oscillations, criticality, wave propagation, and homeostatic balance. And then in artificial neural networks, hierarchical feature representations and attractor dynamics. Okay, and then the last one is resilience to damage, which is in biological neural networks, dynamic rewiring and homeostatic plasticity, allowing for partial recovery. So this is where if you have damage to the system, how does it get fixed or recovered? And of course, this is something that the brain is well known for in terms of plasticity, but we have this sort of, we're interested in how the network rewires itself. So this is famous functional biological neural networks. And then in artificial neural networks, we have static redundancy, things like dropout, and limited on-the-fly recovery. So yeah, I would also add in terms of resilience to damage that there's also redundancy in the biological neural network. And, that also plays a role in recovering information. But there, and then there are connections between these like emergent properties. This area is really interesting, but it speaks to a lot of the other areas as well. And so I like this table, but you could probably keep revising it in terms of different properties and differences that you can think of. But I think the take-home message here is that biological neural networks do things a lot differently than artificial neural networks, and we can learn a lot from biological neural networks in a way that kind of gives us different paradigms of artificial neural networks, at least in terms of that they're sort of even if we had something like an organoid culture or an organoid intelligence, that it's artificial in the sense that it's been sort of cultured and curated by human experimenters, but it's still artificial in the sense that it's doing it's not like the actual biological system, but it looks very different from the artificial neural networks we use today.
I liked that, or it's I'm just seeing that in emergent properties, we've got Beggs, 2008, 2022, and this is the Indiana professor who replicated the mind in vitro, right? So the UIZUC Open Microelectrode Array project, his postdoc is the one that they end the paper with a postdoc was able to build our system in 15 hours and get his own thing running in Indiana, and it's Beggs.
Right.
So it just like that.
Yeah, he's done this stuff with like brain criticality for years, thinking about how that happened. Yeah, thinking about how that happened.
Absolutely.
So and I would emergent properties. I might add it's kind of the brain or neural complexity or brain complexity. So we have all sorts of interesting properties of oscillations and coupled oscillators and things like that. And that provides these very complex dynamics that we don't see in artificial neural networks at all. I mean, sometimes the dynamics are sort of the information processing, but it's kind of that's not going to be captured in an artificial neural network. So I mean, we can come back to this table too. I mean, I think we should collect all these figures and tables, and yeah. Okay, so
for sure.
And I know exactly what week that was that we talked about that other paper with the figure that you showed. It was when we were talking about bioprinting and So I'll go back to that.
Yeah. And he's gonna have a little bit on that too, but yeah.
Okay. Then the properties of functional biological neural networks. So, we're thinking about what are the key features that we need for functional learning capable biological neural networks. Thinking back to if we were going to sort of extract some design principles out of the brain, how do we do that? So the first is neuronal diversity. And that is brain function relies on the interplay between diverse neuron types, including excitatory pyramidal cells and inhibitory interneurons, which regulate network dynamics and plasticity. And so that's, again, where in artificial neural networks you have one unit type. And we usually don't have different types of neurons because we don't deal with things like, different types of ion channels being expressed in the neurons or different types of neural morphologies. We just have the unit, and then the unit has connections with other units, and then they're governed by weights, and that's it. So that's one design principle. The next is modular and hierarchical organization. So, there's this idea that neural circuits are organized hierarchically to produce sensory motor information and execute complex behaviors. Engineered biological neural networks do not fully replicate this architecture. If you look at assembloid or connectoid approaches, they aim to capture key aspects of modular connectivity, but they don't quite get to the level of a mature neural circuit or a mature maybe neuronal network, which of course may be many circuits combined into one functional unit. So, if you're thinking about a network for learning and memory, or a network for attention, or a network for just doing something like addition or mathematical computations, you may need to have different tissues or different brain regions that are hierarchically connected. So, in your typical assembloid, you have a bunch of cell types smushed together, and then they form kind of this functional segregation. But, the organization, of course, is not going to be the same as you see in a biological neural network or a mature one in any case. So this is, again, N of a reach goal at this point, but this is like the one big difference between BNNs and ANNs. The next design principle is long-range communication and critical dynamics. So that's where we have these oscillations like gamma, theta, and beta, and these frequency bands support interregional coordination of memory, attention, and motor control. And then if you're thinking about the dynamics of them, you're thinking about criticality in the brain, balancing, stability, and adaptability. So there are all these like meta features of the brain, different phenomena that are kind of beyond sort of the single cell or even the population analysis. It's, things in the dynamics that we would like to capture for a network, such as what we're trying to do with biological inspiration. Then our final design principle is parallel distributed computation. And of course, there's this sort of area of ANN research that was big in the 80s and 90s, parallel distributed processing, and that's, kind of discovering like parallelism in computation and in computational neuroscience and that. But this is actually kind of thinking about how the brain really takes advantage of this design principle. So the brain's billions of neurons and trillions of synapses support massively parallel processing. We're talking about at that scale. So we're talking about a massive scale and being able to build systems that operate at that scale, but also being able to control systems that operate at that scale. While modern computer architecture such as graphical processing units or GPUs enable parallelization in artificial neural networks, biological neural networks are still far exceed them in connectivity and energy efficiency. So there's this connection between, or at least this thinking that the parallel nature of biological neural networks, the distributed nature of biological neural networks, allow them to achieve these gains that are so far elusive to artificial neural networks. So, energy efficiency being a primary example, but also connectivity and sort of, we talk about these things, it's sort of like magical. It's like, how does the brain do so much with so little energy? Or, there must be some added stuff in the connectivity or something. And we're trying to find like what those things are so we can implement them in some sort of model or device. And so that's what they're getting at with these design principles. And again, there are probably other design principles, but these are the ones they're identifying. Then they're mapping out this NeuroAI spectrum next. Just thinking about all the things going on in the field. This is an interdisciplinary landscape. We have these three domains: hardware, software, and wetware. And, hardware, of course, are things like neuromorphic chips or spike-based silicon systems. The software are computational models inspired by brain function. This could be neurosymbolic AI or reinforcement learning. And then wetware are these living biological neural systems like organoids, cultures, or brain slices. And so you combine these into this these domains. So they also do this categorization of NeuroAI fields, and they categorize SBI or SBI-related NeuroAI into two broad classes. The first is that fields that do not involve living neuronal cells. These are hardware implementations of these kind of biologically inspired systems. So these areas leverage the morphology, physiology, and computational principles of biological neural networks to develop brain-inspired hardware and algorithms. Okay, so this is actually figure 2, a graph representation of the different fields. We'll get into that in a minute, but let's continue with this point 1. And so that the point 1 splits between neuromorphic computing and neuro-inspired learning. So neuromorphic computing is a hardware implementation that moves us away from von Neumann architectures towards things that are more biological inspired. So von Neumann architecture, of course, separates out memory and processing for very practical reasons, but that's not really maybe the way we want to build devices that are inspired by the brain, because that's not really the way the brain works. And so you can build these neuromorphic devices that are much different than the von Neumann architecture. And then neuro-inspired learning, which is where we have different types of algorithms and software that are inspired by the brain. They're not at all like the kinds of algorithms that we have for von Neumann architectures. If you know anything about how to, the different types of algorithmic challenges we have for, like parallel computing and for quantum computing, you know that like the algorithms for those paradigms look very different than sort of the serial von Neumann architecture algorithms that we may learn in like our computer science classes or something like that. So if we have like we have a certain a search algorithm that we build for a serial von Neumann architecture. If we want to parallelize that, it's going to look different. And if we put that on a quantum computer, it looks different still. So there are a lot of these neuro-inspired algorithmic opportunities to build new algorithms, build new sort of computational tools. So the second part of this categorization, so there was fields that do not involve living neural cells, and then 2 are fields that involve living neural cells. So this is where we want to kind of leverage the efficiency and adaptability of brain neural networks and integrate biological neurons with digital systems. So this, of course, is organoid intelligence and bioengineered intelligence. And so this is where you're using organoids or some sort of culture, cell culture, to build these systems that can process information and do this in a way that's similar to what in vivo nervous system does. So sometimes people are using brain slices for this, but other times they're using neural cultures. These BNNs can be integrated with computers to develop effective brain-computer interface systems. So those are the distinctions there.
Yeah, no, just this is my issue with BCI as a term. I mean, it is interesting, right? The Mind in Vitro group has another section on, they first and foremost talk about culturing neurons, right? And again, like, okay, so the easiest thing to do is 2D. The organoids became this new platform because the neurons would self-assemble. I mean, with certain cues, and that added this whole new layer of complexity. Slicing of, taking a brain slice, and trying to keep it alive is definitely another alternative. There's a longer discussion that relates to some organoid intelligence and brain-computer interfaces. This is not getting us any closer to either putting organoids into brains or decoding this isn't decoding a living brain either.
Right.
But these are my issues that I need to work through.
Yeah.
But I love this graph.
Yeah. So this is figure 2. Yeah.
This is cool. And I'd never thought about representing it this way, but it really, it captures well the complex relationships.
Yeah, so this is where we have synthetic biology, cell and tissue engineering, neuroscience, computational biology, and so these are all the fields that kind of go into this enterprise.
Yeah.
And then all the kind of the tools that we have here, neural tissue culture, computational modeling, cognitive and systems neuroscience, machine learning, and then the next level are these kind of different approaches: neural tissue modeling, neuromorphic computing. Then you also have reservoir computing and feedback driven learning, and then synthetic biological intelligence and neurodigital twin model, and then NeuroAI.
I've got there's a lot of acronyms to learn here.
Yeah, they do throw a lot out there.
They're trying to define them for us. But as you go down, they start using their new acronyms.
Yeah, where did they come from?
To make additional acronyms. Okay.
All right. So let's
yeah.
Let's kind of move down the paper. So yeah, they kind of talk about the hardware. Oh, go ahead.
Well, just like again, my only real issue with this paper is that this isn't what NeuroAI means to most people.
Yeah, it's very cultivated for their own.
It's very I can see why Brett Kagan thinks that this is what NeuroAI means.
Yeah.
But when you see all the other NeuroAI papers or like discussion or the what's the Montreal AI? Well, that is AI neuroscience. I don't know. You look at the Patrick Minot NeuroAI for AI safety paper, and honestly, Patrick's probably the most biological of them. But in general, they're more just brain-inspired AI. It's like thinking about transformers as the hippocampus or something like that.
Yeah, I think there's definitely they kind of cultivate this for their own purposes here. But,
yeah.
This is
again, this is my issue. I'm jealous of all this work.
Oh yeah, well, it's great work. Yeah, this is an illustration of a neuromorphic architecture. So this just kind of shows how this is organized without it's very different, underscoring how it's very different from ANNs that we use today. Then they have this idea of off-chip and on-chip learning. This is deep in the weeds of neuromorphic architectures. Then we have, of course, neuro-inspired learning, which are the software models. And then this is where we have neurosymbolic AI and agentic AI. So this difference between sort of a neurosymbolic approach where you take symbolic reasoning and combine it with subsymbolic learning, which is what we use in deep learning or other types of computer vision. But then we also add symbolic reasoning on top of that. And so the idea is you can build structured knowledge, and it's basically combining the best of both worlds in terms of like GoFi versus like the more recent Norwegian approach to AI, which is data driven. And then kind of talking about neurosymbolic AI and some of the ways people do that. This just shows neurosymbolic AI as kind of a model. And then, let's see, they get into the, they were talking about, they were talking about agentic AI. And agentic AI, they don't really talk about that a lot. They talk about it here kind of at the end. Neurosymbolic agents can be made to autonomously interact with their environment, update their internal knowledge representations, and adapt rules through active learning mechanisms. And so this is where they bring up active inference as being kind of the way in which agents behave, and kind of don't talk about it very much. But then they get into reinforcement.
We're going to talk more about it.
Yeah, yeah. So then they get into reinforcement learning and active inference, where they kind of go through reinforcement learning and how that works, and then kind of contrasting that with reinforcement learning and active inference as sort of different ways to do this. Although, there are sort of ways that you can combine them as well. It's just that active inference is really about sort of it's where you're using this measure of surprise, and you're using that to minimize, you're basically minimizing surprise, and that gives you your kind of optimal sort of behavior, or your adaptive behavior. You were going to say something?
Well, just to that point, right? Like the I mean, one, it should be said that Brett Kagan has papers with Carl Friston.
Oh,
right?
yeah.
and so it's just like they're trying to make it sound like, active inference is really adding this brand new thing, and that reinforcement learning needs to have this exploration kind of shoehorned in or something, like I forget the way they put it, bolted on or something like that, I'm not saying it exactly, but it's this paragraph right before active inference. Anyway, but they Ryan Smith does the tutorial, I forget what his paper is called, but it's tutorial on active inference. He's a total active inference guy, right?
Yeah.
Next week there's the, what's it called? International Active Inference.
Yeah, IWAI
workshop.
Yeah.
Is that
yeah, it's the workshop on active inference.
And Ryan Smith is keynote, doing his tutorial thing. And, he's the one who's just active inference becomes reinforcement learning.
Right.
In many cases, that it collapses to reinforcement learning. Yeah. Anyway.
Yeah, I mean,
again.
Yeah, there are a lot of parallels. Yeah, you get the.
Yeah, and I love their figures.
Yeah.
Or so it's fine. I mean, again, they're definitely trying to get at how the active inference model, as described here, is adding something.
Right, and it's kind of. I mean, I wouldn't contrast them, and I wouldn't necessarily say it's because reinforcement learning, you're trying to sort of, it isn't just about policies and selecting policies. It's really about the sort of adaptive behavior. You're trying to find the optimal policy. In active inference, you're trying to minimize the surprise, and you have this, you do have this sort of joint distribution that you're trying to, understand. Yeah. So that's, but they're both kind of very similar, and the structures are similar in that respect. I'm gonna, when we do our presentations to the Active Inference Symposium, I've kind of been summarizing what we've been doing with open source sustainability, and that's one of the themes that we have, is that we have we've done a lot of work on reinforcement learning. We've done some work in active inference, and there's this kind of common theme there. It's emerging, which is that you're kind of using these models to get at some very deep kind of social coordination problems, and how do they kind of get at that in the same way, where what is the structure of these social coordination problems. It's really interesting. I'll have we'll have to go over it maybe next week or week after. Um, but yeah, it's definitely, if you think about like what they're trying to achieve, they're basically converging on the same thing.
Yeah, yeah. It'd be interesting to see. It'd be interesting to expand the current project with a more active inference model.
Yeah. Well, Brian McCorkle was doing that, and he did
Right.
like it was like social active inference. And we think of active inference as being in a brain, but you could do it easily with 8 different agents, multi-agent kind of active inference, and that's kind of a whole nother can of worms. But yeah, we should develop that more. I'm not sure who at the Active Inference Institute. I think that would probably be a good place to go for people to see if people are interested in working on problems like that, working that out more.
Yeah. Yeah. And well, and the other as well as like putting something in the I forget what Daniel Friedman has. He has a particular name for it, but you can definitely advertise projects.
Oh yeah.
with his he just did the quarterly roundtable.
Oh yeah, yeah.
Where he talked about a lot of the opportunities to get involved or propose things.
Yeah, so definitely, we should keep that in mind. So yeah, let's finish this paper up. So we have a diagram for reinforcement learning. We have a diagram for active inference over 2 time steps. And then, we're kind of getting into this table with the differences between reinforcement learning and active inference, just like for different aspects. I don't know if that's necessarily like key to understanding the other part of the paper, which is like we have all these different NeuroAI architectures, because there are other options besides reinforcement learning and active inference. Like, those are 2 methods. There may be other methods that you could employ here, like, or imply that or that you could apply that would be relevant as well. So this is kind of like I guess you could say that reinforcement learning and active inference are based on neural activity, but it's a little bit shady to say that. But anyways, yeah, I mean, I kind of see why they're getting at they're taking this kind of route. But anyways, so this, and then they get into open loop and closed loop learning, which is like where you have this contrast between reservoir computing and feedback based learning. So reservoir computing, of course, is where you have this high dimensional dynamical system. You have these nodes in a container, and they kind of find their own wiring through this sort of this readout layer that maps neural activity to desired outputs. So basically trains the network in a different way than what we see in typical neural networks. And reservoir computing is very hot right now. People are able to do different types of computational things like pattern recognition, line source separation, and time series predictions. These are problems that are kind of interesting to as kind of benchmarks. Not necessarily for like being like doing things like the brain does them, but that's reservoir systems are often assumed to have fixed weights. Biological neurons exhibit synaptic plasticity and may self-organize based on input-driven plastic changes. So these are all kind of, this is one approach, reservoir computing, and then the other approach is feedback-based learning, which is where you have closed loop systems that continuously adapt neural activity through real-time feedback. External stimulation is used to reinforce or inhibit specific network patterns. So just having that contrast between reservoir computing and feedback-based learning. And then, of course, the modular architectures, getting into assembloids and connectoids. So the assembloids are where you have different types of tissues kind of in the same culture. Connectoids, I imagine, are things that like building connectomes in culture. They have so many terms for that. It's like, they have so many terms for different types of organoid. Yeah, so like they define it here. Go ahead.
Sorry. Well, just like yeah, it makes more sense for I get why the assembloid people are doing assembloids, right? Because they care about brain science.
Yeah.
Right? Describing assembloids and connectoids as if they're what's this term? Modular architectures?
Yeah.
Like modular biological neural network architectures? Like,
yeah, it's not really the same thing.
No. It's like, yeah, and what happened to like, I mean, again, like, I'm not necessarily saying they should push this, but like, what about who's the Palm Jeff Hawkins?
Yeah, Jeff Hawkins.
Oh,
right,
like Thousand Brains.
Yeah, hierarchical temporal modeling. Yeah.
Yeah, like I mean.
Yeah, I know they kind of ignore that.
Come up once in this whole thing. It's a review of things. I mean, do they have a beef or something?
I don't know. Yeah, it's like that's.
But just in terms of the cortical column.
Right, right. That would be a logical thing to. Organizational principle, right?
Bring up a cortical column once.
Yeah, I know.
Doesn't that seem like a glaring omission?
It does seem like they're cherry picked for like kind of going in a certain direction.
Totally.
Yeah.
I mean, and this idea of like I mean, as well as like, this isn't even cherry picking, like connectoids versus assembloids, and it has to do with with the engineering, like using, what is it? I mean, I've never seen the term connectoids.
Yeah, that's a new one.
I've seen assembloids that are using microchannel links. Right. Instead of fusing.
Yeah.
Yeah.
Well, let's see. They define assembloids as fused organoids. So region-specific organoids are grown separately and then physically fused together. This is where you get these different.
Yeah. I'm still not exactly sure that's how like Pasca's group or Noblich's
group. Yeah. The idea is that you have. Like, these different tissues that are going to form, and then you have migration, circuit formation, and interregional signaling. Right. So you're just basically putting together a brain from like, Frankenstein's monster brain or something,
Right. I mean, but when Pasca's doing it, right, or Noblish is doing it, they're like, doing a subcortical organoid, a cortical organoid, a cortical organoid that represents, a subcortical organoid that represents this thing, like that, where now I've put together, this this limbic circuit because I want to do a Parkinson's like model or something like that, right?
Yeah.
And anyway,
Yeah.
like again, like I'm not sure why I'm picking on him like this.
Yeah, I know.
But there is some, you know, like it's an arXiv paper. It's obviously not done, you know. But it does strike me as a little bit like the because he was on these papers too. The organoid intelligence paper that had, you know, Thomas Harding and Lena Smirnova, kind of did this same kind of thing. It was trying really hard to like be defining a new field,
right,
you know,
right.
You know,
trying too hard.
We gotta you know, define our terms.
Yeah. So when they say connectoids, they mean microchanneling to organoids. That means that the individual organoids are maintained in separate microfluidic chambers and then connected through microchannels, so they're not really physically together. They're in this set of flow chambers where, you know, you have basically, you can control the growth of each, but they have access to one another. So it's really kind of a limited physiological relevance, but allows you to control the system more directly. And so that's just kind of the difference. And again, you know, these are like different, you know, different methods, basically. And you'd never see that in a real brain. So it's like, I don't know if there's necessarily a point to this distinction.
Yeah.
All right. So I think that's enough for that paper. But that's a great paper. Thank you, Morgan, for bringing it up and having a discussion around that.
Yeah, they've got great. You know, I mean, I think some of their figures are really really helpful, and yeah, you know, like as much as I am picking on some of their terminology, their organization of, you know, the kind of multidisciplinary nature of the work is really, yeah, helpful and I think informative.
Yeah.
You know, and it's very much from a couple people that are like intimately involved in this, right?
Right.
So we can dig around and find what the beef is with them and Hawkins.
Okay.
But there is some more stuff that just goes on that's like super relevant for the dish brain kind of work.
Yeah.
And the not, I mean, you know, the kind of the standard formats and stuff like that. That's stuff that they've brought up before, try to make it look more like a field, but there is a nice section on the recent advances and application, section 10, that's nice because they go over, you know, basically everybody that we've talked about, you know, Final Spark, you know, Braingineers, Indiana, UIUC, Mind in Vitro, you know, and a few others that is cool to be looking up, you know.
Yeah.
And then yeah, and then some like just like their tutorial paper, they've got a great supplementary section. So I'm hoping that this will be, you know, like this will come out next year and it'll be like twice as big, and you know, really being kind of like the defining, you know, is like what I'll be pointing students at.
Okay.
You know?